NIH Cambridge program, Wolf Lindwasser and Riddick Chowdhury, found that at least some of these acidic clusters also bind to a subunit of the AP2 complex, the alpha subunit of AP2. And finally, you are probably all very familiar with the ubiquitination of lysine residues in the cytosolic tails of transmembrane proteins with functions as a sorting signal for internalization and targeting to the MVV pathway and to lysosomes. This is something that we did relatively little work on, but at least for some of these proteins, again, Rosa Puertoriano found that ubiquitinated cytosolic domains are recognized by another domain, the gut domain, of the same 3 gigaproteins that we had discovered before. Okay, so here we have now, like 15 years later, a compendium of signals and adapters that mediate sorting of transmembrane proteins in the endosomal lysosomal system. You can look at it as a dictionary that allows you to read the language of protein sorting in the cytosolic tails of transmembrane proteins, and in many, though not all cases, the presence of these signals allows you to predict how they are going to traffic within the cell. So where do these interactions take place? Well, they take place at different stages of the endosomal lysosomal system. Interactions with AP2, ARH, and Disable2 take place at the plasma membrane and mediate rapid endocytosis of various endocytic receptors, transporters, adhesion molecules. While interactions with the other adapters take place in intracellular sites, and they mediate different sorting steps between endosomes, lysosomes, and related organelles. Now, all of these machineries are extremely important. They are present in all cells, and mutations, as Owen mentioned, in either the signals or some of these adapters, the recognition molecules, are the cause of various genetic disorders. And years ago, in collaboration with Bill Gall, we showed that mutations in one of the subunits of AP3 were the cause of a pigmentation and bleeding disorder known as the Hermansky-Purlak syndrome type 2. And in addition, again, as Owen mentioned in his introduction, pathogens hijack many of these interactions in order to enter cells and to replicate within cells. And we've also done some work with funding from IATAP on the mechanisms by which the NEF protein of HIV hijacks AP2 in order to promote rapid endocytosis of the CD4 protein from the surface of cells. Now, there are a lot of interesting stories that I could tell you about each of these adapters and each of these interactions, and we've done work on all of them. However, for the remainder of my talk, I would like to focus on the AP complexes. And in particular, I'm going to show new data on a new AP complex known as AP4. The AP complexes are composed of 4 subunits. They are heterotetramers. They have 2 large subunits, an intermediate-sized subunit known as mu, and a small subunit known as sigma. These 3 complexes are composed of subunits that are homologous to one another, and they all have the same overall structure of a head with 2 ear-like projections, 2 ear-like domains. This structure has been likened by some to a Mickey Mouse head, except that this is not a very good structural model for the AP complexes, because Mickey Mouse doesn't have these long hinge-like sequences that connect the ears to the head. But it sort of helps you remind you what these complexes look like. Now, these complexes are ubiquitous. They are present in every cell in our body, and in all of your favorite cells, T cells, fibroblasts, neurons, hepatocytes. They are present in all cells. they are clathrin associated. They use clathrin as their coat, as their scaffold. And they are recruited to membranes not by interactions with the signals. Those are too weak. But they are recruited to membranes by virtue of interactions with members of the ARC family of GTPases and by specific phosphoinositides, specific lipids on each membrane. And the use of different ARC proteins and phosphoinositides confers a different localization to each of these AP complexes. Now, as I said before, AP complexes recognize two types of signal. Remember that? Tyrosine-based sorting signals, YXX multi-hydrophobic, which we show bind to the mu subunits of the AP complexes, and also dilute signals, which bind to a combination of two subunits of the AP complexes. So there are two different binding sites for each of these types of sorting signals. The structural basis of these interactions are now well known from the work of David Owen at the University of Cambridge, who solved the crystal structure of AP2 or its subunits in complexes with these signals. Now, we know quite a bit about these three complexes, and I'm not going to tell you about everything we've done about them. However, a few years ago, another postdoctoral fellow from my lab, Esteban de la Angelica, who is now an associate professor at UCLA, and I actually lifted this picture from his website. He looked younger then. And so he discovered the existence of four adapter protein complexes. And this discovery, the same as the discovery of AP2, which was also discovered by Esteban de la Angelica, was a direct product of the ESP sequencing project and the genome sequencing project, because that's how we found proteins that were related to the subunits of the two known complexes, and we then demonstrated, we put all those subunits together into these two complexes. So Esteban discovered AP4, and this complex is different from the other. It's structurally related, but it's different from the other three in that it's not associated with clathrin. And in addition, after the discovery of AP4, we hypothesized that perhaps AP4 will be able to recognize the same type of sorting signal as the other three complexes. So we tested for interaction of AP4 or its subunits with various tyrosine and dilute-based sorting signals that we had in the lab, and we failed to demonstrate any interaction. So it was really different from the other three. And eventually, we got frustrated with this complex, and we stopped working on it because we couldn't find any signal that bound to these complexes. And a couple of other labs did some work on AP4, but eventually the field forgot about this complex for a few years. And it was not until recently, and a little bit serendipitously, that we finally found a cargo molecule that interacts with AP4 and that is sorted in an AP4-dependent manner. And this is what I would like to tell you about now. So this cargo protein that interacts with AP4 happens to be the Alzheimer's disease amyloid precursor protein, or APP. So how did we find this interaction? Well, it was a little bit by accident. Two postdoctoral fellows in my lab, Fabi Burgos and Gonzalo Mardones, who are here in the audience, were actually working on a different project. They were trying to demonstrate interactions of APP, the amyloid precursor protein, with the retromer complex, which is another signal recognition complex on which we have done some work. Because the group of Scott Small at Columbia University had found that certain sporadic cases of Alzheimer's disease presented with reduced levels of retromer. And he went on to demonstrate that knockdown of retromer resulted in increased production of the amyloid beta peptide, which is the pathogenic agent in Alzheimer's disease. So we were interested in determining whether ATP interacted with it. But we couldn't find those interactions. So we failed, again, to demonstrate any interaction of ATP with the retromer. But in the course of that work, Hari and Gonzalo had the idea